structure and meat quality. And we're going to go through it based on a few questions. I always think it's better with a presentation to have questions that we're trying to answer so that we take note of the main points. We're going to look at what muscle fibers are made up of, why muscle structure is important when we're producing meat, what affects eating quality, and then what can we do to try and affect the eating quality. As a presentation, I've got some questions for you on quality, tenderness, flavour, juiciness of the meat, and the colour of the meat. And to make it a little bit easier, there's those that are in red on some of the slides. So if you have a pen and paper ready now, you can down your answers before we even get there. So moving on to the first section, muscle structure. Most of livestock animals is normally between 35 and 60% of their body weight or their carcass standard weight, which is fantastic because that's our yield. Obviously, there's a variation in animals there, dependent on the condition of the animal coming in. The muscle mass, what about muscle mass as a whole? We're actually talking about muscle fibers, connective tissue, the adipose tissue or fat tissue, the vascular system, so the blood supply to the muscles, and the nervous tissue, so the nerves that go to those muscles to make them move. Okay, actually 3 different types of muscle in the body. There's cardiac muscle, which is the most reliable, which is used in our heart. There's smooth muscle, which is the muscle that's in your esophagus or your intestines. It's the muscle that provides that peristalsis movement through your gut, where that aids your digestion, and is the twitching that you see on the green offal during post-mortem, because it does continue to respond at post-mortem too, once the animal's stunned. So the muscle that we're looking at that makes up the meat is skeletal muscle, and that's made up of 90% muscle fibers and approximately 10% connective tissue or fat. And if you look at the these different types of muscles, both as a diagram or under a microscope, you can see that skeletal muscle, it's got these striations through it. Smooth muscle all fits together around it, and then cardiac muscle has these areas that interlink, and it's these connections that make cardiac muscle more reliable. Muscle fibers are obviously made up of muscle cells as well. Each of those cells has proteins in it. It has actin, myosin, myoglobin. They're all different sorts of protein, and they're the ones that are of most interest to us today because they look at contraction and the relaxation of the muscle, and the oxygen carrier is myoglobin, which affects the meat colour for it, the amount of oxygen that's in the meat. The cells also have in them water, minerals and vitamins, fat, both in and around the cells, and glucose or glycogen, so that's our energy source. So look at the structure of muscle. This would be your muscle, whether that's a primal, whether that's your own tricep or bicep. So the muscle as a whole, each of your muscles is made up of fibers. And each fiber has muscle bundles with it, and each bundle has an individual myofibril. And that is then made up of the different muscle cells. So that when we cut through, it provides us that green in it. Because each of these bundles as well, and each of the fibers and the muscle itself all have connective tissue around them. This area is going to be important as we start moving on to looking at muscle contracts and relaxes. We mentioned that actin and myosin in the previous slide are the proteins in the cell that help it contract and relax. And the cell which runs down the center is important because that's the straight line that allows the muscles to contract and relax around it. And the important point is that all of this is surrounded by cytoplasm, which allows it to move and slide around. So, muscle contraction. I have simplified this because there's lots of reactions, lots of different compounds that can, some have very long names to them. Basics of a muscle contraction is that the brain sends a message through your nervous system to say, move your arm. So that muscle. The nerve then sends a message to the muscle. And that message is forwarded to a sodium compound. It releases that sodium compound, releases a calcium compound or calcium ion that tells the muscle fibers, so the actin and myosin proteins, to move over each other and contract. And that's done, so it triggers movement. And movement is done a bit like a rivet structure, where you've got a rivet over a tooth structure. So it's stepping along and in place through these little sections up here. The blue line would move over the red line. The same point. Distance between these two outer, what they lay, is shortened. So if we look at this second photo here, the blue stays the same, and the pink moves over inwards, and then here reduce in size, so you end up just with a band in the middle that stays the same. And muscles doing that contraction, it uses what we call ATP, which is energy or glycogen, or glycogen to ATP, and there is what we mentioned before, through the message to tell the different proteins to move over each other. Here we've got 1, 2, 3, blue ones, and then the 6 once again. So it's that over each other. Whereas in relaxed muscle, we've got 6 here and 6 here. It means that it's not as contracted and compressed together. Unfortunately, it's quite difficult to get good photos of muscle contraction that are free from copyright, but you can go Google searching and find some that might be able to explain it a little bit better. The structure, how they step over each other and cross over. So this is another picture that shows relaxed muscle at the top, and then once again we've got the red and the green overlapping in your connective muscle cells. Luckilation is a lot easier because the brain is stopping all of that messaging. So it sends a message to stop the movement, the nerve stops producing the signaling compound in the calcium, reabsorbs that, and the muscle, the trophomyosin, which is a different form of protein, comes over that riveted structure. So the myosin protein slides back out to their original position, making the muscle longer and more relaxed again. So moving on to the next form of protein to look at within a muscle cell is myoglobin. Myoglobin is important because it's what gives the meat its colour. It's a water-soluble protein and comes in 4 different forms. So we can muscle or meat in bluey purple colour when we just freshly cut it. We can see it in a red colour and we see it in a brown colour. And each of these different forms, deoxymyoglobin, oxyglobin, carboxymyoglobin, and metmyoglobin, produce different colours. And the best learning meat colour is this triangle. And it's called the meat colour triangle, where once you've fresh a piece of meat or a primal, you'll see this blue-purple, which is the deoxymyoglobin. So no oxygen has managed to get into the globin yet. It's just been cut open. Once it's cut open, there's then 2. It can weave it in normal air and it will bind to oxygen and bloom, is the term used in molecular chemistry. So the oxygen or the myoglobin holds on to the oxygen and becomes oxymyoglobin. It's that lovely red colour that we like. And the other option is that when we vacuum pack meat, for example, carbon dioxide binds on to the myoglobin and we still get a red colour, but it's called carboxymyoglobin. There is, however, a third option, the option where meat goes brown. That normally happens because this carboxymyoglobin action can reverse and the myoglobin can let go of the carbon dioxide. It can go back to the blue, but it quickly changes to one of the other genes, either oxymyoglobin or metmyoglobin, or if not enough oxygen in the air and the meat has taken up as much oxygen as is around it, and then there's more oxygen in the air around it than is in the meat, it actually starts seeping back out in air to try and make the 2 equal. And the meat can either go to carboxymyoglobin if there's carbon dioxide in the air, but that doesn't very often, or it can go to the metmyoglobin, which is where there's any other compound with what we call oxygen radical, a free oxygen compound, such as hydrogen peroxide, will deoxymyoglobin and turn it this brown colour. So when we're packaging products, we need really to understand this triangle. Put it into vacuum pack. You take all of the oxygen out of the product, have an environment where there's nowhere for it to react with, and we end up with the carboxymyoglobin. We can in matte packaging or your fashion kind of cling wrap, glad wrap over foam tray with any air in there, and it will start off as this oxymyoglobin, but potentially change over time, especially with the matte packaging, so over time into this myoglobin form. Then if you, and that's the advantage of matte packaging, is that if you put a pressure to the air or you change the makeup of the air in the container to be oxygenated, you can keep it in this oxygen state, oxymyoglobin red state, longer. So all of this is done by myoglobin, which we said was a protein, and there's enzymes that changing between the different colours and the different forms of myoglobin. It can also be affected by pH and also by temperature, so affected by the environment around it. I touched on this previously, whether the myoglobin can lose its ability with storage. And it's because enzymes break down and as the meat breaks down, but that's long storage, a lot of time. From the scientists I've spoken to and heard talk, made it clear that the pathway to oxymyoglobin is good, but as soon as it releases back to deoxymyoglobin, it's actually normally easier for the myoglobin to bump with something else, and that's why it would go brown versus go back to a red colour. Unless associated with oxygen, that is. It's important to understand this triangle too, because when people are shopping for meat, they're obviously looking for this red state normally, and there's perception that the brown state is actually because the meat is going off versus what's actually happening, which is colour chemistry. It's not to do with quality or the food safety of the product, it's more to do with the colour chemistry at times. And there's a reference down the bottom here that is from it's a guide to meat colour. It's a very good book and it's freely available on the internet if you go searching for it, or contact myself or Mintrack and we'll give you the link to it. The other part of muscles and muscle that's really important is the connective tissue. And there's two different forms with muscle. There's collagen and there's elastin. So collagen is formed through length to stabilize and strengthen the muscle. And we did say when we were looking at the muscle structure that there's connective tissue around the bundles, around the fibers, and around the muscle itself. So the tip around collagen is that when you cook meat for, it will soften into a gelatinous kind of, whereas unfortunately with elastin, which are mainly your ligaments between bones, that don't actually break down, and that can be really tough, which is why we don't tend to, Australia anyway, eat tendons, because they're elastin based and collagen based. Okay, so we're going to move on to now, after talking about the structure of meat, onto meat quality. And we quality in a number of different ways. So we can talk about quality of nutritional quality of the meat. We can talk about quality for the high quality, the food safety, microbiological load on meat. But in this case, what we're going to do is talk about eating quality of meat. And we use MSA, so Standards Australia, and their parameters around eating quality. And it's scoring out of 100, with broken down into tenderness, juiciness, flavour, and overall liking of the product, and has gone through a lot of research with consumer testing and sensory testing of meat. So there's a number of different areas that can affect eating quality. Through each of these individually. So there's the function and growth and weight gain of the animal. There's the marbling of the meat, the amount of glycogen the animal actually has prior to processing, the age and maturity of the animal, the hazards used for the carcasses, and aging of meat. So quiz, and you'll see there's some lovely words in here, so answering the questions at the end. Start with finishing growth and weight gain of the animal. Obviously, this is an area that is more important from a producer aspect than for to look after. You can ensure your buyers are buying animals in good condition. It's predominantly a producer area for them to look at to get the best yields and the best grading of their product. So in animals that are finished to the best conditions, so high quality feed, lots of feed, you end up with the optimum amount of muscle and intramuscular fat, which makes the product and makes the meat more tender, increases the flavour of the meat, and results in it being more juicy. And if you when just from your own point of view, when you eat well, you eat a lot, you eat calories or kilojoules, we all, unless you increase your exercise at the same time. So if you're having a excess of intake, it allows you to put muscle on, it allows you to put fat on. But if you're not getting the nutrition you need, what your body does is break down fat normally first, then breaks down your muscle. And breaking down your fat, it leaves the muscle and it leaves connective, and it doesn't have any fat to break down, and then it starts breaking down the muscle, but the connective tissue still remains there, which means that you increase the proportion of that connective tissue to the muscle or to the fat available, and that results in a tougher product. So it's important that in that last stage, that finishing section, that especially those 2 weeks leading up to processing, that enough feed, good quality feed, they are still laying down muscle and fat cells to get the best product. honestly, especially in Australia where we've got varying weather and rainfall conditions, ability of feed, producers always have a choice to be able to provide that good quality feed. What's shown that in that 2-week period, there isn't a difference though between a grass or feedlot fed nutrition, but we'll move on to that a little bit later in the presentation. So on to marbling. Marbling helps the juiciness of the product. When we talk about marbling, we're not talking about real marbling of fat around the muscle as a whole, which is about the intramuscular fat deposits. So both around the cells and the cells themselves. And around a prong, for example, or the layer of fat around the carcass. Fat laid down in an uneven way through the body, and it increases towards the end of the body and decreases towards the tail in livestock. So cattle or any animal has to be on a high nutritional plane, gaining more energy than they need to maximize that marbling ability. And marbling can be affected by things. It can be affected at the beginning of the animal. So if there's been good enough nutrition early on in the animal's growth, they don't seem to have the same capacity to down that intracellular muscle, intracellular fat, as well. And also 2 weeks leading up to process. The other areas that can affect marbling is genetics. So breed, we all know Agues are well known for their juiciness and their level of marbling, but also individual animals. Going back, they genetically have the ability to lay down more intercellular within the muscle cell fat than any breed. Within animals, we tend to look and go, if I process if I produce my animals, if I feed them the same, if I give them the same preventative health, wormers or vaccines, if they get the same water, the same shelter, I handle them all the same way, then I should get them all to the same grading. But just like if you put 50 people in a room and gave them the thing to do or the same amount of food or drink, there would still be people in there that were hungry or thirsty or did the task what I asked them to do a different way. And that's different in animals. So in your or in herd of animals, you'll get ones that do a little bit poorer and ones that do a little bit better. And thing would be to get the majority of them to that grading area, into the marbling area of what your specifications are. Okay, one of the areas that affects the quality of the meat is muscle growth. So glucose is energy glycogen and are energy reserve in muscles, and this affects the pH of our meat and the colour of our meat. It increases with feed, and it can take weeks to increase with feed. So feed is broken down into simple sugars and stored as glycogen. When it's not used and there's excess, that's when we get the fat deposits for our marbling. But it very quickly with stress. So innovators, the recommendation that they're on good feed for 2 weeks prior to processing, and that's because of this glycogen level. It takes a long time to build up. The most you can possibly get of in the cells and in the body, but it can decrease very, very rapidly. When we're talking about glycogen, we talk about a glycogen bucket. Everybody has that, each animal has their own bucket. It can be big, it can be small. And in feeding them good quality feed to processing, what you're hoping is that you've got that filled way up to the top as far as it can be. And it affected, the amount within that bucket can be affected by stresses, whether that's handling stress, transport stress, climatic stress, and where both producers and processors can make an effect on the animal and the quality of the meat. Obviously, a juicer that can fill that bucket up, but through the ledge, through the transporter, we can affect the amount that's in there. Glycogen in meat is really important because glycogen gets broken down normally into lactic acid. In an animal that's alive, that lactic acid gets broken down and the animal keeps going. It's enough when you sprint and you get a stitch, it's because of that lactic acid, or when you get a cramp, it's because of that lactic acid. But in because it doesn't get broken down any further, and the glycogen converts to lactic acid, and it's that lactic acid that reduces the pH of the meat. For a pH in the meat around 5.3 to 5.7. Oxygen in the animal, more lactic acid, the lower pH goes. So it's important to have that glycogen bucket as full as it can be so that you can reduce the pH and prevent dark cutting. We call dark cutting. So this is pH, and there is another on understanding pH, Contraction and ANPC. We'll go over this quite quickly, but if you've got more questions or want more information, I recommend you go back and have a listen to that webinar too. pH, what we're actually looking at is the effect of the pH temperature and what we call the pH window. And the reason the pH and the temperature go together is because they both have the ability to change proteins and the enzymes for how rigor mortis or that muscle contraction happens. For an ideal temperature window where the carcass goes through rigor mortis and pH is 6. And if it's that pH temperature window, it means we'll get the best possible eating quality. So as the drops with the converting to lactic acid and dropping the pH in the meat, we want to make sure that the carcass is also at an ideal temperature. And the best way of showing that is through a couple of graphs here. So we're not saying that we can prevent dark cutting or the product, or we can't cold shortening of the product. We're not saying we can prevent dark cutting through this, but what we can prevent is the cold shortening and the heat shortening, where the muscle contracts too quickly or too further than we want to get a less touch. So this is your pH window, and as you can see, the green line goes down, pH over temperature. That means we get the best quality we could possibly have got for that piece of meat. If in pH lower, the temperature decreases quicker, then we up with cold shortening. And reverse, if the pH drops quicker to the temperature, then we get a heat shortening effect. And a similar graph here for beef. The benefit in this graph is that with different hanging methods, whether it's an Achilles hung or a Tenderstretch, you can have a different pH window. So, questions: what can we do? pH temperature window. Obviously, more glycogen input, but based on the production affects how much goes into that bucket, and based on the individual animal for how big the bucket is, but also on the producer for how well fed the animals are in those 2 weeks prior to processing. And both the producer, the transporter, and the processor can affect the glycogen output or the holes in the bucket by reducing the stress, reducing the handling of the animal. So make a difference with using electrical stimulation, and we change the temperature for that pH window. So looking at time of processing, the time on the floor to floor, chilling temperatures and chilling methods that you use. Okay. Another area that can affect the meat quality is the animal, and affects the tenderness. So, animal ages, the fibers in the muscle tissues get stronger, they get more rigid, and they're not broken down as easily through cooking. So that results in the meat being tougher. But at age, we can talk about it in a number of ways. We can talk about it as years, if you know what the animal is in years. We can talk about it through dentition, as in approximation of years based on how many teeth have come through. Or we can talk about it from from a physiological point of view. So this is based on ossification and is around how much those cartilage areas have matured, and cartilage can clear down and compress into more of a bone structure. And that's with age. So then gives us a different measure for physiological age, because it can also be affected by the nutrition and the development of an animal. And we do hear people saying, oh, you look good for your age. And this is the sort of thing we're talking about, where nutrition and the life that the animals had can make a difference in their physiological age. It can be affected by genetics and hormone balances, as well as the nutrition. That shows the difference in a younger animal, where there's separation between the vertebrae, than an older animal where you've got fusion areas into more of a bony structure. Okay. So where processes can definitely make a difference is in the hanging method used. And these are more used in sheep than they are in beef. And methods used. So there's the Achilles method, which is over on the left here, where the carcass is suspended by the Achilles tendon, or the Tenderstretch method, where the carcass is suspended by the pelvis. So hanging meat, what we're doing is changing how rigor mortis occurs, or to what extent rigor mortis occurs. And your animal to allow both the weight of the carcass and the tension to pull the carcass down and prevent some of that contraction. By preventing that contraction and the laying of those proteins, as we saw in how muscle contracts at the beginning of the presentation, you prevent a toughness in the meat. And forms Achilles affects the entire carcass when it's doing that, but it doesn't apply evenly or as evenly over carcass versus the Tenderstretch method, where the legs are dropped to 90 degrees and allows more of a stretch over that hind quarter, so that you get less contraction. So it has a more tender stretch. Hanging has more of a positive effect on the hind quarters. But whether we're talking about sheep or beef, they use more, the two different variations use more in sheep processing, and less in beef processing because of obviously available space in chillers. Dropping that to 90 degrees takes up a lot more space and therefore ends up costing a lot more money, which is comparable to the increase in price that you can get through the quality of the meat, is my understanding. So the two different hanging methods compare each other. Obviously, dependent on the time to boning, it does show that they both reach the same point. For example, if you boned out at this here and you were using the Achilles method versus the Tenderstretch method, you would potentially get a better tenderness from the Tenderstretch methods and the Achilles. But as we're going through this presentation, we're showing that there's a lot of different things that can also affect the tenderness of meat. I did in here connective tissue and cooking. MLA spend a lot of time on educating consumers, which is fantastic. Depending on the connective tissue and then the method you use to cook the meat, so different areas of our body have different amounts of connective tissue around them, dependent on how much we move and use those muscles, which then results in how long you need to cook them for. So, for example, a foreleg, which is used for movement of the animal all the time. This is those back muscles that, yes, they're used for stability, but they're not active or relaxed as much as your foreleg. Will produce your nice tenderloin stretch, and your forelegs for sheep, lamb and is fantastic, but it's used through a slower cooking method, and that's to allow the time, a lower temperature and longer time to break down that connective tissue into, as we described earlier in the presentation, as like a gelatinous texture. So it's important when we're producing meat and when we're cooking meat to ensure that the consumer understands some of the structure or the meat for how to cook them, get the best results with the product and get the best texture. And that's saying the thing, sorry. Including, for example, we use some of those cuts with more connective tissue in them that we use for casseroles or meals. Okay. Another option for us too is aging meat. So when you're eating, you're looking more at a microscopic level. All of the area that we were talking about in the beginning around how fibers contract and relax and the muscle structure, because over time the enzymes naturally break down. So those proteins start to naturally break down and it weakens the muscle fibers, so meat becomes more tender. So aging can be done both in carcass form, it can be done in primals, it can be done in vacuum packaging. Obviously vacuum packaging or even other forms, we just have to be sure you've got the food safety right to ensure the product isn't affected that way too. Lastly is packaging. So depending on what packaging you've used, that can affect the tenderness of the product, and as in meat color, it can, and the meat color triangle, it can affect the color of the product. In the news in October, I've just some articles here that came out at the back end of last year around packaging, and the fact that through research done in Australia, they were actually looking at cutting of meat, is my understanding, but they certainly realised that packaging can also have a negative impact on the tenderness of the product. This has also been found in sheep meat in New Zealand as well. So it might, if you're interested in that, going back and having a look at those Beef Central articles. There's one here on impact, and there's also a response from retailers on that. The AT are looking at adding deduction with MSA on MAP packaging, but before doing that, MLA are going to do some more research to see if there's ways that you can prevent MAP packaging having that impact. Obviously, retailers and processors have invested a lot of money in MAP packaging, and it would be very difficult to reinvest more money in a different form of packaging. So it's good that they are going to do that research and try as a way of mitigating the impact. Coming to questions. If you have a pen and a piece of paper next door to you, let's see if we have, hopefully you've jotted down some of the answers as we've gone. Let's see what you can come up with, answer the presentation for what affected juiciness. And what I'll do is give you a minute or so, and I can go through some of the answers for you. So guess what affects juiciness? Please, you've written down because that's cellular fat that results in it being juicier. And because the marbling is based on the nutrients at the end of the animal's life, you popped in there as well, nutrition, juicer end. So what is it? So everyone managed to get an answer for this one because the majority of what we've gone through today actually affects tenderness, whether that's meat, the age of the animal, so the ossification, the way the meat's hung, the carcass is hung, where the carcass gets any electrical stimulation to prevent or reduce that rigor mortis, the feeding of the animals, one thing to ensure that there's a high glycogen level to reduce that rigor mortis again, to decrease the pH, that basic temperature can have an effect because of that pH temperature window, and packaging, as we saw at the end of the presentation. Possibly. Next one is what can affect colour. Remember colour triangle too. The colour triangle, remember, is around oxygen levels. It's around the length of time the meat is out to be able to bloom or to be in different environments, and the packaging can affect that. There's glycogen levels because that can change the pH, temperature parameters, and affect whether that meat ends up dark cutting or not, and if there's not enough glycogen, it can be dark red, which means it's darker despite that triangle. So the amount of stress the animal is under can also be an answer to that one. The next round, flare effect. This is a bit difficult. But have it. Even a presentation to see if you can find it. A little bit more difficult, as I said, and flavour can depend on the intramuscular fat based on the type. Our last question, and here's some of the references we used when we're putting together this presentation. This top one, the American Meat Science Association Meat Colour Measurement Guidelines, is very good, and as I said earlier, freely available if you're interested in meat colour. So if you want to get in touch with myself or Track, we'll be able to provide you a link to that if you can't find it through search. Any questions? Touch with myself or Mintrack, and we'll help if you can. Thank you very much, everyone, for your time and having a go at the questions. And I'll hopefully see you on soon. Thank you.
Thanks, Elizabeth.
Thanks, Rachel.